Hi everybody, and welcome to the Malone Lab project flowchart. This is going to be an overall generalization of what our lab does without the nitty-gritty of each step. So let's start at the beginning. Everyone starts with choosing their gene. They also, as soon as they find that, right, they must find their gene in the database with their sequence and design primers to the promoter region, or the putative promoter region, which sits just upstream of the 5' UTR of the RNA sequence within the DNA. PCR of this putative promoter region will be generated and then subsequently cloned into the PGC Blue PCR cloning vector. Now, as you can see, there's red arrows coming off of this design primer step. If you were also using this as a timeline, which we will all do soon, you can see that these items over here are done simultaneously with these items over here. These are wet lab, these are computer lab. So there's always something to do while you're running a gel, waiting for PCR, waiting for cells to grow up, or what have you. So let's go down this row first, and then we'll come back over here. But as we mentioned again, these should be done simultaneously. We do not need to wait to get down this row in order to start down this row. You may want to not start immediately, especially if you're brand new to the lab, to make sure you're concentrating on your one experiment at a time at the beginning. Soon, however, you'll be able to multitask with the best of them. So back to PGC Blue, we clone our putative promoter in the PGC Blue vector, and then we subclone it or move it from one vector to another, the PGL3 clone, which is also our luciferase vector. This is the final vector that we want our promoter in to drive the luciferase gene to make the enzyme to cleave the substrate to give off light. That's our readout of promoter activity. So this, the PGL3 basic, is where we want our promoter. This guy's just an intermediate to help us clone this much more easily. We do a transfection to make sure we get some activity, and then we design our deletion constructs or our truncation constructs to find the most important region of our promoter. We do transfections of all these deletions. When we find the most important region, we then mutate the putative transcription factor binding sites in there and do more transfections. Well, how do we mutate transcription factor binding sites? How do we know where they are? What's over here? Again, simultaneously, we should be analyzing our sequence using computer databases and algorithms to find potential transcription factor binding sites. We have a list of transcription factor search sites that you can put your sequence through that will give you a compilation of all the putative transcription factor binding sites within your promoter deletion constructs. We don't necessarily care about binding sites in regions that we find are not important, but we can start with the whole sequence, get our putative binding sites, and then narrow it down based on the data from our deletion constructs. At the same time we're doing this, we should be finding all the orthologous sequences of our promoter. What's an orthologous sequence? All the other species that our gene is conserved in, and then we're looking to see if in the conserved genes is the promoter sequence also conserved. We do a multiple sequence alignment of the promoters to look and see how related they are, narrow them down to the most related, and then look for conserved transcription factor binding sites across orthologs, which will help us determine which transcription factor binding sites we should mutate. So not only do we do a truncation construct to narrow down what transcription factor sites we should mutate, we also do an evolutionary analysis to again narrow this down. We don't want to have to mutate 10 different sites. We want to mutate 4, 5 at the most. So these two methods help us narrow in on what transcription factor binding sites we should mutate and transfect. That gives us function. And function, or how your promoter works, right, is what we then, what? Publish! Yay! Right? That's essentially what our main goal is at the end, yeah? Well, you guys graduate and we publish. You guys go on to bigger and better things. As a bonus, we'd like to do some other experiments if we have time. Why? Because we can get a better publication. Woohoo! If we have extra experiments. What do these entail? EMSA, looking at the transcription factor binding sites and what proteins actually bind to these, or do a protein bind to them, and which one is it? CHIP, or chromatin immunoprecipitation, is looking at those transcription factors binding in the chromosome itself, not just in vitro in a test tube, but actually in the chromosome of an endogenous cell. We can also do an RNAi against these transcription factors, or whatever transcription factor we think might be the most important, right? Knock it down in the endogenous cell, see what happens to the expression of our promoter or to our endogenous gene. And we can also overexpress these transcription factors to, again, look at the function. Does this increase our promoter? Does it increase the endogenous gene? Right? Give us more information, more stuff to talk about, better publications. That's the big picture, people. Let's get some data. Thanks.